as Secretary General of the International Union of Basic and Clinical Pharmacology and Editor-in-Chief of Pharmacological Research, which is a journal affiliated to IUPHAR. We have developed this idea of joint seminars between the International Union and Pharmacological Research family of journal to specifically answer to one need, which is the need of young investigators who are at the beginning of their careers to build their own scientific path, and they may need a little bit of help in terms of technology, methods, and collaborations. So this series of webinars is intended to provide these young investigators with basic tools in pharmacology and the possibility of connecting with the scientists, senior scientists who are giving the talks for collaboration and possible mentoring. This is now, I think, the 6th seminar that we are having in this series, and we would probably be happy to have your feedback on this series of webinars, whether they are good, whether you have any suggestion to them. So feel free to contact us. And I think we will organize in the second half of the year a meeting with you all to have your feedback directly. So I'm passing now the M to Anne-Marie Pordon.
Okay, I'm going to share a couple of slides here. There we go. Can you see my slides? Yep. So my name is Anne-Marie Pordon, and I'm a pharmacology publisher with Elsevier. I'm delighted that you could join us today for the July webinar for the Pharmacology Education Initiative, which Emilio has introduced so beautifully. We'll be taking a break from webinars in the month of August, as so many people go on vacations during that time, but we'll be back in September. During the break, we're going to be working hard to move our webinars to a platform called Cassini, which is dedicated to webinars and seminars, with features such as being able to give a webinar a DOI so that it can be cited. Also, it'll be a single place where we can announce future webinars, store previous webinar recordings, and also build a community around the webinars. We also hope that with this platform that we'll have fewer issues with platform stability. We've had some issues with Teams in the past few months, and we want to improve that. Before we begin the webinar, I wanted to inform everyone that you will not have access to camera or microphone during this webinar. Any questions should be entered into the Q&A at the top of the screen, and we will answer them after the webinar. Now, I would like to briefly introduce our speaker today, which is Associate Professor Duangrudee Tanramluk. She's an assistant professor at Mahidol University's Institute of Molecular Biosciences. She earned her PhD in biochemistry from the University of Cambridge in 2010. And her research is very interesting. It integrates artificial intelligence and structural bioinformatics to accelerate drug discovery. Her expertise includes quantum chemical calculations, protein crystallography, and software development. She also developed a web platform called Monora.org, which she's going to describe later in the site and during the session and show you how it works. And this platform supports in silico drug discovery by linking ligands to proteins, genetic data, and biological systems. She also designs algorithms to analyze conserved protein features, aiding in inhibitor design. Her innovative computational tools aim to make drug development faster, more efficient, and cost-effective, contributing to advancements in healthcare. So, with that introduction, I'm going to stop sharing my screen once I can figure out how to make that happen. There we go. And Duangrudee, now you could put up your presentation and on with the show.
Okay, it's a real pleasure to be here to talk for the Pharmacology Education Initiative and for IUPHAR. So I will give a little webinar on the arts of drug design, and I hope that it will inspire younger generation scientists to do drug discovery using computational tools combined with wet lab experiments. So I want to start now, so let me share my screen. So the outline of my study is about the basic of structural biology, the drug target analogy, traditional structure-based drug design and fragment-based drug design, case studies, and novel methods from my lab. The rational drug design consists of several fields of expertise. First, we need to obtain the structures from X-ray crystallographic study or cryo-EM or NMR. And then after we got the target enzyme structure, the computational chemist will do the drug design analysis. And then the organic chemist will do inhibitor candidate synthesis. And then it will pass through biochemists to do in vitro analysis, pharmaceutical chemists to do in vivo testing, and then it will undergo pharmaceutical chemistry testing. The whole process takes around 10 to 17 years per one drug and costs more than $1.3 billion US dollar or more than $2 billion US dollar in some cases. So my work is in computational biology or bioinformatics at this step. Before going deep down into the algorithms, I would like to introduce a little bit about basic structural biology so that you can keep going with the rest of the audience. So the paradigm for structure-guided drug discovery, first we need to have a gene of interest, and then we scan through all the biological space to find the new targets. And then we get a target protein like this, and then we screen the whole chemical space at either in-house or at drug company in order to find a new drug or an inhibitor which inhibit this protein. So the basic of protein structure, first the protein will have amino acid, and the string of amino acid letters will have repeating torsions and become alpha helices or going on this way and this way like this and become the beta pleated sheet. So they have the repeating torsions, and then if the combination of them become alpha, beta, alpha, or alpha, alpha, we will call it a motif. And if it have a recognizable folds, it will be called a domain, such as solenoid, clamp, sandwich, or horseshoes. And then if it have functional form, it become tertiary structures. And if they have cooperativity among them, like this one in hemoglobin, we will call them quaternary structures. If we put the atom into the periodic tables, it will have various representation styles and colors and various radii. So in visualizer, you can see that you will have the color like this, and for amino acid knowledge understanding, we will use this row. So the color to memorize is carbon in white, nitrogen in blue, oxygen in red, and sulfur in yellow. And for molecular representation, if you are a chemist, you probably draw the line like this. And when you become a drug design researcher or crystallographer, you will draw the atom in ball and then the bond in cylinder. Or if the radii of the ball and cylinder are equal, it becomes the stick representation. And then if you draw it according to the relative radii of van der Waals radii, you have the CPK space field, which more clearly resemble the vicinity of the active site. So this one, glutamate 86, histidine 87. So something like this. And when you want to see in structural biology, you omit all the side chains and see only the main chain. So the amino acid side chains consist of 20 amino acid letters. This is the 3-letter code and this is the 1-letter code. The 1-letter code for glycine is like this one is very small amino acid and it allows flexibility in the protein. And then if it becomes alanine, valine, leucine, or isoleucine, you can see that they don't have special colors and they serve mainly the structural purpose. For serine, threonine, and tyrosine, you can see one red tip like this. It allows phosphorylation or other post-translational modification. For cysteine, it becomes reducing agent and allow for disulfide bond formation, which you can alter it for thermostability, for example. And if it have two red tips like this, aspartate and glutamate, it become negatively charged, partial negative charge, I would say. And if it have one blue and one red, it become polar uncharged, as in asparagine and glutamine. For lysine, arginine, and histidine, you can see that it have blue colors. Lysine and arginine, you can see it in tryptic digestion, or people talk about these blue colors amino acids in epigenetics, for example. And proline introduces king in the helix, and phenylalanine, tyrosine, and tryptophan mainly cause pi-pi stacking hydrophobic interactions. So all these ones are crucial in certain way because of the character of them in the amino acid in the protein. And for protein representation, you can see that you can have alpha tracing, which you can see clearly how many units there are and trace along the main chain. And for ribbon or cartoon, you can see secondary structure. For schematic, you can see arrows or rocket for beta sheet or helices. So these ones you can see the direction of the secondary structure. And for van der Waals surface, it more truly resemble the true shape inside the pockets. And this one is the representation. When you use cartoon, you basically, if you draw the visualizer, you will just draw a thicker line along the main chain like this, and it becomes alpha helices. And if you omit the side chains, it becomes these structures. And schematic, if you zoom closely, this one to go from N terminus to C terminus, from N terminus to C terminus. And for van der Waals surface, it looks like this. You have the charge character, positive in blue, negative in red. And when you design something to attach to it or inhibitor, you basically try to find the positive color to attach with the negative colors. So you want to find the opposite colors, for example. So the keys to the design: suitable size, shape complementary, and charge complementary. I want to help you understand the target first by my little analogy of cat. So let's make analogy. If the protein target pocket is a cat's face, you can see that this one is the HIV protease. It looks like a cat. You can roughly say it looks like, but you haven't dissect it to how. So let me see this one. How can they look like this one? So it have 2 eyes, 3 whiskers, 2 ears like this, and it have face like this. And so the protein pocket looks like a cat face in this analogy. So we have this protein and we have this look like a cat. And then if you want to design, assuming inhibitor or a drug molecule you want to put a mask on this protein face like this. And you want to make the mask perfect fit on the face and then stop it from biting, for example. Now, this is the sample sets that you have. This one is sketch, this one grumpy cat, smudge, orange cat, yellow cat, exotic short hair, and Siamese. So if you are a drug design researcher, how can you choose which target you want to design something for? So sketch is probably not a good input because it have low resolution. Grumpy cat and smudge is really multi-targeting. It has exists in various social networks, and it will go viral easily by designing something for it. For orange kitty and yellow kitty, these two from similar origin and they looks very similar, and it's really easy to miss out something. So this one is what causes drug side effects. And for Siamese and exotic short hair, if you want to design something for it, it will be specific, unique, and really customized for their face. So the structural quality is very important. For 2D models or the models generated ab initio or something, it's really hard to tell where the side chains are. But if you have three-dimensional X-ray structures like this, it's probably much better. So the input data is the very important part before you do any experiment. You have to make sure your input data is very good and high resolution. For off targets, as you see a lot in kinase inhibitor, it merely from designing the inhibitor for something and in the same time it attached so many other ones which have similar origin, like in homologs of them or something. So these ones is off targets. So we don't want these ones, so it's become off targets. But if we want the variation like this one, so it become pan-variant inhibitor. We have this mask for Persian, for exotic short hair, for Scottish fold, and for Egyptian. So this one is the idea why I interested in studying pan-variant inhibitor design, especially for COVID-19. So again, the key to the design, suitable size, shape complementary, and charge complementary. So what make a good drug targets? Almost all drug targets are protein, and it should have known mechanism of action. Specificity is essential, so there should be no similar other essential molecule. And there should be druggable, so it should have the binding size of the size and type that can be targeted by small and tractable molecules. Question about your target. For target identification, the drug researcher will ask which protein are good targets for this disease. For target validation, we merely ask whether they are druggable. And for lead identification, whether they have the right activity at the chosen target. And for lead optimization, we want to look at which variants have the right pharmacokinetics property and also non-toxic. So in designing, there is a lot of molecular forces that affects binding affinity. The first one is covalent bond, which the bond that share electron pairs between ligand and target atom. And then there's van der Waals interaction, which is weak and non-specific attractive forces caused by transient dipole. For ionic interaction is the attraction between the cation and the anion. And for hydrophobic interaction is the result of aggregation between nonpolar molecule that cannot hydrogen bond with water molecule aggregate together. For hydrogen bond is the interaction between hydrogen atom in a molecule with a partially positive charge and another atom with unpaired electron like this. So for molecular docking, which people have done a lot. It's based on lock and key concepts of pharmacology. The aim was to find molecules with suitable conformation by using the thermodynamics free energy terms to filter the possible pharmacophores of the ligand. So basically, they have the protein and then they remove all the water and then prepare the protein with the correct protonation state. And then they try to design something like this to find the correct orientation and correct location. So the protein like this, you can see side chains, and this is the cleft of the van der Waals surface. So the energy reduced from minus 5.6 from red colors to orange color to yellow color to green colors. So the aim was to try to have the lowest score, and then they rank around like 10 or 20 of them, and then the top performer ones, they will guess that it can predict whether or not it binds in the real systems. But it's not a true promise whether it will work, and the score is not related to real physical chemical properties. It's just an estimation of binding interactions. And for fragment-based drug design, if you have the pockets like this in semicircle, triangle, and rectangle, you will design something with suitable size and suitable shape and suitable charge. And then you basically grow them, link them, and then merging them. And there is also linkers and bioisosteric replacement, where you replace the chemical compounds, chemical substituent with the similar ones in physical chemical properties and try to optimize pharmacokinetics properties or toxicity or solubility or whatever. So I want you to listen to Sir Tom Rundle, who is a pioneer in the field of fragment-based drug discovery. His lecture is very inspiring, and let's and share the screen.
Give me just a second while I get this up. Oh. Can you see my screen? Yeah, okay,
Yes.
I'll start the video.
Against a target, so you need to use other techniques, some physical techniques. So what we proposed was that if we knew the active site of a drug target, and we could imagine that there were 3 sites, and we had a library of 1,000, we could explore those 1,000 molecules to see whether they bound there, there, and there with the same library. But we'd have to detect them with biophysical methods or X-ray methods to see where they went. And I thought in 1997-8, what we would do would be to cross-link them all. In fact, it turned out that the best way was to grow them. And so then what happened is we proposed to venture capital that we would have an experimental system, we'd have known structures, and we would screen those with a very small library using high throughput biophysical and X-ray methods. And sooner or later we will get one that binds, although I sit here sometimes wondering whether I will, and there you are. That's a fragment bound. You wouldn't get it in a biological activity, but you can spot it and define it using the sort of methods, X-ray methods, NMR, and many other methods. And then we get chemists in to elaborate it. And so that's the general idea.
Okay, thank you. So let me go back to my slides. So after we get the fragments bound, these hits will be undergoing what we call the hit expansion. So it have high affinity towards the target, normally less than 1 micromolar, and it should have selectivity versus other targets, significant efficacy in cellular assays, have drug likeness as in Lipinski rule of 5, good binding affinity and high cell membrane permeability, and high water solubility as well. And it should have low interference with cytochrome P450 and P-glycoproteins, low cytotoxicity, metabolic stability is high, chemical stability is high, and it should have synthetic tractability, and also it should be patentable. So you can see that the whole process is really laborious and require a lot of money, a lot of experts, and a lot of resources. Let's have a look at the case study. Let's say there is a myth about depression, about chemical imbalance. So I will try to explain why we end up with the drug like citalopram, for example. So depression affects millions of people worldwide. The way we combat is by trying to find the target in the brain cells called serotonin transporter. In the brain, there is axon and dendrites, and at the end of dendrite, the axon have the end called synapse. The synapse have serotonin transporter here. So serotonin naturally expressed in the brain but produce much lower quantity in people with depression, and a drug like citalopram inhibits serotonin transporter and blocks serotonin from being reuptake back to the nerve cells. So it leads to increased concentration of serotonin in the synaptic cleft, and people believe that it will make us feel happy again. And in animation, this one is the video thanks to Jacqueline Mason, who created this video. So this is the brain cells, and at the end this is synapse. You can see serotonin transporter here, and serotonin being reuptake back in red molecule. By blocking this transporter with inhibitor like this, so the serotonin become sitting in this junction longer, and they believe that the depression will get better by this process. And if we want to look at the structure of serotonin transporter, it will have the structure as the follow. So the transporter look like this. If we look into the pocket and plot by pKa, we can see it looks like this, and this is citalopram. And it will have positive charge bind with negative charge attack with positive charge, and the hydrophobic patch between the two rings like this. So this is the real life example of what it looks like. And activity is not enough. Most drugs that fail in clinical trials are active against their targets, and failures often due to difficulties in administration, drug concentration, drug-drug interactions, or drug side effects. These are called ADMET issues, or absorption, distribution, metabolism, excretion, and toxicity. So the question is, how can the drug be administered? Normally, oral route is preferred. There is no use that if you invent some very nice drug, but it need to be injected one time a day, for example. So normally the small molecules is the aim of our group, because it's very easily transferred to a rural area and can apply to wide range of people. And how is it distributed in the body? For CNS compound like citalopram, whether it will cross blood-brain barrier is very important. It needs to be able to cross the blood-brain barrier to get to the brain cells. And how is it metabolized? This can affect drug-drug interactions, such as the bad example one is the ergot, which cause more than 100 drug interaction. And by treating migraine with ergot, the patients may end up have leg amputation and arm amputation because of the drug-drug interactions. And will it cause unacceptable drug side effects? So for application of computer-aided drug design, when the target structure is known, If the potential ligand structures are known, we dock ligand into the binding site or do structure-based pharmacophore. If no potential ligand structure are known, we will do de novo ligand design or fragment-based drug design. And when the target structure is unknown, whether AlphaFold is usable is still questionable because the accuracy at the side chain level, whether the mutation, only one point mutation will affect the binding or not, that's still a problem to be discussed. And we can do QSAR, but that's not the gold standard for it. So I would like to introduce my platform called Manora. Manora is in short for mapping analogous nucleides onto residues and affinity. It's a machine learning platform to guide protein and ligand design by anchors and influential distance. So Manora tried to link chemical biology with structural biology through its algorithm for binding affinity and distance correlation. And we have the dissection of molecular interaction and protein and ligand conservation. And then we have big data backend to link sequence structure, RNA baseline expression, and target organs. And we link to genetics and systems biology to SAMU and KEGG with hope to do drug design better. The paper was published in Structure by Cell Press and has been one of the most read papers at their website. So the underlying principle of my approach is from computational chemistry, merely quantum chemical calculations. So when I did that work of finding the potential energy surface of cycloguanil, I found that they normally talk about the preferred lowest energy conformer using density functional theory. So there is the conformation that has the lowest energy, and the gold standard for them, whether their trace is a good energy structure or not, they will trace it back with comparing to X-ray crystal structures. So I also continue after that work to explore the statistical analysis of thousands of amino acid environments which show frequently occurring atoms. So you can see the structure of tyrosine like this. If you have the structure into 20 amino acids and then dissect it into each class having one same amino acid as the central atoms, and then if you superpose large enough of them, you can see frequent patterns of environment. So if the drug molecule is this puzzle piece, what I try to study about this red piece is by superposing the environment like this, a lot of them, and try to find the piece in the middle, this one. So how can we know what is the missing piece? My approach is in trying to find influential distance that correlates with KD and anchoring points, which are this point, and then frequently occurring atoms, basically dissect it into classes of carbon, oxygen, chlorine, nitrogen, and others, and try to find frequency of it. And then I did the active site boundary to understand the pocket from unionizing all the ligands possible in the pockets. That time I did with the structure of MPRO, the main protease for COVID-19. So software symphony is basically having the structure of protein in the middle, and then we put it on the grid, and then we try to find the environment with various grid size. And from what I found, this one is the conserved atoms and residues. If you superpose a lot of kinase based on this set for adenine atom arrays and this set sterosporine atom array, you can see that the highest conservation is different. For the inhibitor, the conservation can go up to 75% or more. But for the natural substrate, it's pretty much more flexible. So this one is the concept of induced fit. So this atom is induced towards this position with large amount of structures in the studies. And the value that we try to correlate it to the structures is this one called the binding affinities. So at equilibrium, if we put the protein and ligand in the test tube, we can measure what we call the dissociation constant by multiplying the product between protein and the ligand and divided by the protein and ligand complex. So we want the complex to be with high concentration and we want the KD to be very low. If we want the KD small, we hope that it means the ligand can bind very tightly. And then we generate the distance matrix. So we measure the distance in the pocket of the protein kinase. And then we measure the distance in the matrix like this and tabulate it in Excel files. And then if we sort it 1 PdV per row and on the column we have the equivalent distance from each central atom of the amino acids, we can see that if we sort the last column by the KD, the tightest bound to the loosely bound have measurable parts that are often very large distance or often very small distance. And if we do the regression, we can see that if we want to lower log KD, we can dissect it to distance term like this. So this one, distance between amino acid residue 50 to 184, if it have positive coefficient, it means the lower is the better. And if it have negative coefficient, means the larger is the better. So with this way, we can tailor-make the molecule like this. So for precision design, we have the constant parts, we have the expansion parts, and then we have the contraction parts, and we have the parts that we still don't know because it have amino acid identity far beyond the distance alone. So with methoxy and methylamine, I put it onto the platform that I create. So this one, if you find the hydrogen bonds from this atom N4, you can see that if it have 2 hydrogen bonds and the Ki will be quite small. If you have only 1 hydrogen bond and the gatekeeper is large, the Ki is medium. And if in contrast it binds to this O6 oxygen instead, the Ki become 9,000. So it doesn't like to bind to these oxygen atoms. In contrast, it like these nitrogen atoms. And for active site boundary of the binding site, you can see that this one is the puzzle piece that consists of many little pieces of the smaller ones. So basically the idea was in COVID-19 era, there is a lot of X-ray crystallographic structures available. Within 2 years, there's more than 300 crystal structures. So we superpose large amount of crystal structures to find the unionizing of the ligand in the pocket. And this is what we found. If we unionize all the ligand atoms, we can see it look like this. And this is called the active site boundary. And we can dissect it further to atoms that is more frequently than the others. So this atom in Paxlovid is nitrogen, but in fact, when we did the calculation, we found it to be carbonyl carbon. And we prove it with molecular dynamics and MMPBSA. So In conclusion, Manora have crystal structures as input and link out to protein data bank, open targets, Campbell, SAMU, and cake. And then we find the conserved structures like this. And then we have the anchoring points. From the anchoring points, we measure the influential distance and interresidue distance. And then we hope that our approach can lead to ligand modification and protein engineering. And then in the future, it can help drug discovery easier and with other applications to be discovered in the future. So we prove it with X-ray crystal structures. So from our PDB code that we crystallized to solve the differential distance equation, we found x, y, z coordinates to be like this. From this one, aspartate 54 to threonine 185, and we find the distance like this. And this distance, if we crystallize the structures and then we put the distance inside the equation of influential distance calculation, we can calculate the Ki to be 0.95 nanomolar. And if we do experiment, it's got to be 0.2 nanomolar. So it plots very nicely on the log Ki MTX with various DHFR species. So the influential distance look like this. When you put it into the structures, this one is the distance. And we can use this influential distance to find the value of binding affinities. So for the basis for tailor-made drug design, we have the conserved part, which are position-specific interactions by symphony. And we have the variation parts correlates with the Ki, which are distance, either expansion or contraction. And we have interaction types, which are dissected using Manora. And active site boundary in our website called mprocovid, which aggregate the information from more than 400 crystal structures to get the precise design of atomic positions. And we have the demo, but since I'm already here, maybe I demo it using the live platform. The platform consists of several experts. We have Professor Jisnu-san, who is the earliest supporter of this project, and we have crystallographer, system biologist, crystallographer, kinetics person, molecular biologists, several IT people and database people and distributed computing people. And we have computational chemists, database and bioinformatician. So an all-in-one platform and wet lab and dry lab integration to tailor-made drug design will be the key to tackle AI-driven drug discovery problem. And I will demo for like 5 minutes, and then I will be happy to take questions. Let me stop sharing, and then I will demo the platform. Share. Windows. So the platform can be accessed from manora.org. Then you can look up any chemicals. It can be used as molecular drawing tool for you. So let's say trimethoprim like this. You can look up. Seems like everyone access at the same time. The panel hasn't coming up. Let's go by the other way. So let's go to sample. Basically, it can draw any structure that you want, and then you can access. The fastest way is to PDB 3-letter codes. So in here, you can put MTX like this, and it have methotrexate like this, or you have top like this for trimethoprim, and then you can superpose atoms that you want, like this one, and then you can do drug design analysis. So the panel look ยังไม่ไปหรอ
share window
ต้องแชร์ window โอเค share screen and then drug design analysis like this. So you can see it have the very fast way to structurally align the structures, and then you can see that it have influential distance equation like this one from B2 to B9 leucine 5, which I mutated to valine, and it gets better binding affinity from negative coefficient like this. So we can look into the structure. We have the structure like this. We have the distance, and we have the contraction and expansion distance. And then you can look into the folic acid biosynthesis. Now you look into the folate biosynthesis pathway like this. And then it can map the DHFR 1.5.1.3 into this pathway. And further than that, it can even go to organ and tissues. So you can go to open targets like these ones. And you can see the baseline expression. So the RNA is expressed in immune organ and in blood, and the protein expressed only in immune organ but not in blood, for example. And let's see other functions. You can have protein and ligand interactions. So let's look at our case study, staurosporin like this. So I show you the example of using N4 and O6, and then you choose all these structures and then choose protein and ligand interactions. Now it have the structure for you to load, and then it have binding affinity. You can see the hydrogen bonding. This is one hydrogen bond. You go to another structure. This is another hydrogen bond with different distance. And if the binding affinity is less, you can see 2 hydrogen bonds like this one. So you can see that the application is vast, and you can see the COVID-19 platform that we built. This one, SARS-CoV-2, we superpose 300 crystals. And these structures, you can see which atom is more frequently occurring than the others in COVID-19 main protease. So you can choose the atom types. Let's see, nitrogen, like this. You can see which nitrogen is more important than the others. And you can see the line representation for all the ligand combines. So you see the density of all the ligand combines, unionizing density of them. So we have 10 minutes left. I will be happy to take questions. Thank you.
Thank you so much for that fantastic webinar. So yes, we'd like to open up for questions. As mentioned, you can put your questions into the Q&A, and we look forward to receiving those.
While we wait, can I ask a question, Liv?
Yep, of course. Go ahead, Emilio.
So my question is as a very simple one. I like your presentation, and it's very straightforward to me. My question is, how much can you predict the success when it comes to biology? I mean, you can create all content, identify molecules, bondings, and things like that, but when it comes to biology, is that a significant improvement over what we have been using so far as a sort of predictive model?
You're muted.
For the biological systems, we basically have the protein that bind to the crystal in the bind to the ligand in crystal, and then we find the protein pathway based on the X-ray structures. So it means this ligand bind to this protein, and this protein exists in that cake pathway. And then we link the protein to the baseline expression in open targets. That means the protein expressed in that tissues and in that organ. So if the ligand end up to be at that tissue or that organ, it's gonna be binding to that protein. So it link all the database together, and it have biological meaning from the traceable pathways.
Very clear, thank you. So in essence, we are saving a lot of time and a lot of bench work. Is that correct? By using your tools. That's great.
Samia, go ahead.
Yes, just while others are coming with questions, first of all, thank you so much. It was excellent, very clear presentation, very, very good for the pharmacologists that are not so familiar with drug design from your perspective. So it was excellent. My question is perhaps very basic, but I'm wondering the case, for example, when you work with crystal structures, you are, if I understood correctly, you are working more like with a static structure. And I'm thinking how much we can extrapolate that in a case where we have flexible conformations of the target, or it changes the conformation according to the environment. I can give a case if the question is confusing, because I work with a protein that the conformation changes according to the content of cholesterol in the membrane. So what would be your approach? You have to design several crystal structures of each conformation, or does it make sense what I'm asking? Sorry.
So basically you chop the ligand or the part of that you want into the small ones, and you try to focus on the part that you can see. And it's not the tool that solve all your problem, but it's gonna tell a little bit of, if you want to understand its fragments, you search through the fragments and you see the preferred interactions that occur for the whole protein data bank. So with 100,000 crystal structure, the data set is good enough for you to start discover something using intuitive approach. So it's like a calculator for drug design researcher. So I cannot answer from the top of my head, but if you have your query, you plug it in and then you see what it turns up, and then it guides you through various ways, like interactions that is preferred, or maybe it will tell you about the target that people discover it from crystallizing this cholesterol with other proteins, for example, and it saves time for you to perform all the wet lab experiment. So it have the educated guess for you. So as a tool builder, I try to make it useful for drug design researcher, protein engineering, and pharmacologists.
Thank you.
Thank you.
We have some questions in the Q&A now. We have one that says, thank you for the nice presentation. What do you think about de novo design of potential inhibitors with ML-based approach?
Universal design of the, say again, please. I cannot hear clearly.
Yep. What do you think about de novo design of potential inhibitors with ML-based approach?
The novel design of potential with ML base. So for ML, I use data that exists. So for de novo, it's gonna be non-existent. So the whole de novo is not achievable, but each fragments that build up to them, we can search through the database. So you can have part A, part B, and part C. Each part you can search through the platform and see the environment, and then you map it together.
Okay.
The machine need to learn something. It cannot predict anything it never sees before. It has to base on some data, some existing knowledge, and we use the PDB as the training data to have high quality data sets. Does that answer your question?
We have another question as well. What would be the best methodological strategy to start in vitro testing after initial design and synthesis of a small molecule, SPR? If it's not clear, you can also click on Q&A and read yourself as well.
Oh, okay.
It might be helpful.
Q&A. What would be the best methodological strategy to start in vitro testing? So in vitro testing, you should try to find whether or not it's actually the real target, right? Or do you want to actually synthesizing the molecules? Can you say more clearly?
Wait and see then if there's a follow-up response.
What would be the best methodological strategy to start in vitro testing after initial design and synthesis? There's various ways. You can find the KD, right? You can find the KI. You can find... Does that answer your question?
Okay. Danny.
Yes. So thank you. It was really, really interesting for us that we are more experimental biologists and pharmacologists. And I learned a lot. And I already know that is probably very difficult to determine the crystallography of a protein. And I believe even harder when we talk about receptors, especially the ones in the membrane. So I would like to know how hard it is, and if you have available in your software, in a platform, Manora, if you have receptors there, protein receptors, and if you have an experience on studying this, with doing docking, studying with small molecules and receptors.
It's very hard to get crystal of membrane protein, very hard. And if it exists in protein data bank, we have it. If it's not, we don't. But I used to perform docking experiment for the membrane protein like ion channel, and it gives nice results, but I'm not sure whether it's applicable to all other cases, but in some cases it works very well.
Thank you.
Okay, we've got some more in the Q&A. There's a question wanting to know if your group is open to scientific collaboration with groups from Europe, and if so, what projects might be of interest to you?
Group. Yeah, we open for collaboration in Europe.
You know what? This reminds me actually that I'm going to share a slide that has a code for getting a certificate, but it also has an email address that can be used if somebody wants to contact us after, and then I can put you right in contact with this person.
Okay, I forgot to share the last slides. I'm sorry.
It's okay. No, it's okay. I have it too.
Let's share the last slides.
Oh, no. I had one up that has that information, but it also has an email address.
Yes.
Let
this
me
one?
share my. No. Oh, well, yeah, could be. They could contact you directly there.
So this is my email, and this is my LinkedIn, and this is my website. And can you capture this? You can Duangrudee at Hotmail or duangrudee.tan@mahidol.ac.th, or you can reach me through LinkedIn, or you can see the websites of my achievement. You can see mprocovid.com or manora.org. And this one is the claim code for those of you who want the certificates.
Excellent. And I'm also putting that into the Q&A about the code. And then we have a couple other questions that have come up. Do we want to go a little bit further? Do you have time for that? Okay.
Let's see.
The question is.
Stop sharing. And Q&A.
One from Sabrina.
Sabrina. Sorry, I asked in the chat, didn't see the QA. Can we use your platform and add an already synthesized molecule to change it? My platform is available at manora.org, and currently it frees at before the AI era to avoid junk in, junk out. So you can upload your crystal to the website, and then you can analyze your in-house crystal if you have the structures. But for the synthesizing molecules, we only offer what's in there. We cannot, you cannot build your molecule in our platform, but you can search through fragments. Each query takes quite a while because it searches through so much amount of data, but it allows you to search through the chemical drawing and small strings. So you can have the structure that you want to design in fragments and then search each fragments. It's not gonna have you the chimera of things because it only exists existing data in the protein data bank. So you can see fragments of things, but you cannot combine it to unicorn and hope that it's gonna show you the unicorn. You just see the horse, you see the cow, and you cannot see unicorn.
Okay. There is another, so let me know if you want to keep going. We can also ask people to send their questions into you directly. The question is, what do you think about the use of biomarkers to monitor drug response during clinical trials?
I think clinical trials in large parts depends on the patent safety at the end. So I would try to focus on the effects on the real biological systems rather than relying on the biomarkers alone. I am not expert in clinical trials. I'm just a tool builder. But if I use the best of my knowledge, it should be done to the best of patent safety and biomarkers sometimes cannot relate to the safety at the end. Maybe
So we're going to stop there. Any questions about collaboration? Of course, you can write directly about that. So we'd like to thank everybody for attending the webinar, and we look forward to seeing you again in September when we're back with more. Thank you. Bye.